Diffusion. What is diffusion? What affects the rate of diffusion? How does facilitated diffusion differ from diffusion? And this is part of the 3.2.3 transport across cell membranes topic of the cells AQA topic. So we want to look at simple diffusion involving the limitations imposed by the nature of the phospholipid bilayer, and facilitated diffusion involving the roles of carrier proteins and channel proteins. So what is diffusion? It is a form of passive transport, and it is the net movement of molecules or ions from a region where they are more highly concentrated to one where their concentration is lower until they are spread out evenly. And the rate of diffusion depends on the concentration gradient. So the greater the difference in the concentration, the faster the rate of diffusion. The area over which diffusion takes place, so the larger the surface area, the faster the rate of diffusion. And the thickness of the exchange surface, so the thinner the exchange surface, the faster the rate of diffusion. So this is known as Fick's law, and diffusion is proportional to the surface area times by the difference in concentration divided by the length of diffusion pathway. So for fast diffusion, you're going to want a large surface area, a large concentration gradient, and a short diffusion pathway. And diffusion is also affected by the nature of the plasma membrane, the composition and the number of pores, and the size or nature of diffusing molecule. So small molecules, polar molecules, and fat-soluble molecules diffuse faster. An example of a surface which is adapted for a fast rate of diffusion are epithelial cells which line the small intestine. And they have these microvilli, which are finger-like projections or folds in the membrane. And then they give the epithelial cell, a larger surface area. They increase the surface area by about 600 times. So the larger the surface area, the more particles that are exchanged in a short time, and the faster the rate of diffusion. So facilitated diffusion now is still passive, so it doesn't need any energy. It relies on the inbuilt motion of the diffusion molecules. So things that can be facilitatedly diffused are glucose, amino acids, chlorine, calcium, etc. And they still go down a concentration gradient from high to low concentration. And there are specific points across the membrane where proteins are present, some of which can form protein channels which form pores in the membrane for charged particles to pass, like chlorine or calcium, to diffuse through down their concentration gradient. And without these proteins, the diffusion would be very slow. Now these protein channels are very specific, so there are different protein channels which facilitate the diffusion of different charged particles. So these protein channels can control the entry and exit of certain substances. Then there are carrier proteins, which move larger molecules, such as glucose, down the concentration gradient. And they are specific, so they have different carrier proteins which facilitate diffusion of different molecules. So glucose, for instance, will have a specific glucose carrier protein. Amino acids will have a different specific carrier protein. So what happens in this process is, first, the large molecules attaches to the carrier protein in the membrane. The protein changes shape and it releases the molecule on the other side. Okay, we just want to look at the role of diffusion and absorption, which we've already touched upon earlier on. But the net movement of molecules from high to low concentration, that's what diffusion is. And as carbohydrates are digested continuously, there's usually a greater concentration of glucose in the small intestine than in the blood. And obviously blood is constantly circulated by the heart, so there's always that, or there's usually a diffusion gradient. If there isn't, it would then have to use active transport. So glucose is removed from the blood when cells use it for respiration. And villi are specialised finger-like projections in the ileum, which line the ileum, and they contain muscles that contract and relax, mixing the contents of the small intestine, which have new glucose-rich food which replaces it, and it maintains a concentration gradient for diffusion. However, as said in the active transport video, diffusion can only result in an equal concentration at each side, so not all glucose can be absorbed by facilitated diffusion. Some of it will have to be done by active transport. So let's just go back. I just wanted to say something because this comes up quite a lot. So these finger-like projections here with the muscles, with the lacteals in, that is a villus. Then if you look at the epithelial cells on the villus, these brush-like borders here, they are the microvilli. So the microvilli are what are found on the epithelial cells, and you can see it again here. They are the microvilli. Factors affecting the rate of diffusion. The rate of facilitated diffusion depends on the concentration gradient. So the higher the concentration gradient, the faster the rate of facilitated diffusion. But however, as equilibrium is reached, the rate of diffusion will level off. The rate of facilitated diffusion also depends on the number of channel or carrier proteins. So the number of channel or carrier proteins, if there's more of them, it'll increase the rate of diffusion. However, once all the proteins in a membrane are in use, once they're all occupied, then it limits the rate. So the facilitated diffusion can't happen any faster, even if you increase the concentration gradient, because it is the carrier or channel proteins which are limiting the rate, or the availability of them. So the greater the number of channel or carrier proteins in the cell membrane, the faster the rate of facilitated diffusion. And I just want to go back to the fluid mosaic model. So use the fluid mosaic model to explain membrane properties. So the membrane is a good barrier against water-soluble molecules. So phospholipids are a major component of the bilayer, and they have hydrophobic tails which make it difficult for water-soluble molecules such as sodium ions and glucose to get through. And the membrane controls what enters and leaves, and that's because protein channels and carrier proteins allow passage of large or charged water-soluble molecules that would otherwise be difficult to cross the membrane, and different cells have different proteins. The membrane allows cell communication. That's because they contain receptor proteins and they allow the cell to detect chemicals released from other cells, which signal the cell to respond, like hormones, like insulin binds to receptors in living cells, and this communication is vital for body function. And different cells have different receptors. The membrane allows cell recognition, so some proteins and lipids in the membrane have short carbohydrate chains, like a glycoprotein or glycolipids, which tell the white blood cells that the cell is self. The membrane is fluid, and that's because there are phospholipids in the plasma membrane which are constantly moving, and there's more unsaturated fatty acids in the bilayer. The more unsaturated fatty acids in the bilayer, the more fluid it becomes, and the more cholesterol, the less fluid it becomes. So if you got given a question like this, use the fluid mosaic model of membrane structure to explain how the membrane controls what enters and leaves the cell. It was worth 5 marks. Here would be the answers you would give. So the hydrophobic tails of the phospholipid bilayer prevent water-soluble molecules from diffusing through the membrane. Protein channels and carrier proteins control which of these water-soluble substances can enter and leave the cell.